Welcome everyone to this mini lecture of Advanced Topics in Biological Research. I'd like to give you a little bit of background information regarding epigenetics, imprinting, and I'm going to be using the example of the Igf2r gene locus. So what is epigenetics? If we were to look at the word itself and to break it down based on the origin of the word, it means on or above the genome. It's genetics beyond just the sequence of DNA bases, right? The sequence of nitrogenous bases. So this is an additional layer of information that affects the expression of genes. And this is done by modifying the genome. So in eukaryotes, we can actually define the genome both as the DNA molecule itself, but also the histone proteins that the genome is associated with. Remember that in eukaryotes, DNA is packaged around clusters of proteins called histones. So we can modify those histones such that the DNA is more tightly packaged and thus typically repressed because other proteins can't access the region of DNA anymore. Or the histones can be bound more loosely, or the DNA less tightly packed together. And now this can open up the region to expression because now other proteins that facilitate expression of a gene can access that region. So we can chemically modify the DNA or the histone proteins in order to affect gene expression. Typically with DNA, the change that we see is methylation, particularly of cytosine bases, although we're not going to get into that level of specificity. But methylation of DNA is typically going to repress a specific region where the methylation is occurring. So the DNA becomes more tightly packaged, this is one possibility, and thus gene expression is repressed, or at least that region of DNA, whatever its function may be, is going to be repressed. There are a variety of chemical modifications we can make on histones, including methylation and acetylation, and the effect of these chemical modifications really depends on where in the histone cluster the modifications occur. So methylation and acetylation can happen in a variety of places on this cluster, and where and in what pattern the modification occurs is going to either enhance or repress gene expression in the region. We're not going to get to that level of specificity, but suffice it to say that it's really complex, and it turns out that there's this whole code of chemical modification that you can look at the pattern of modification of the DNA and of the histones and predict whether you're going to shut down a region or you're going to open it up to be used by other proteins. So we're going to focus on DNA methylation specifically, since we don't have time to go exhaustively through the entire epigenetic code. But DNA methylation usually occurs in clusters, again on cytosine residues. So if we were to look at a small region of DNA that's say 20 or 30 base pairs long, we would see multiple cytosines within that region with this methylation modification on it. So it usually occurs in little patches in the genome. And depending on where in the genome this happens, there can be a variety of functions. Since we've added these chemical adducts essentially onto the DNA, it now has a different shape and polarity, and now it can act as a platform for other proteins that affect gene expression. So it can affect how an enhancer works, it can act to recruit other proteins to the region, all sorts of things. It can also, the DNA methylation can act to enhance histone binding. Again, if this were to happen, you would recruit more histones to the region and the DNA becomes more tightly packed around these proteins, and we essentially crumple up this region of the genome so it's very tightly packaged up and becomes inaccessible to other proteins. You repress whatever function that region of the genome has. And this pattern of DNA methylation is heritable, both when a cell divides, and so the daughter cells are going to have the same pattern of methylation, and it can be heritable across generations. So when one organism produces offspring, there's a methylation pattern that is inherited. So in terms of the cell cycle, remember that part of the cell cycle involves DNA replication. And so as the new daughter strands of DNA are produced, there are enzymes that can come in there and also copy the methylation pattern from the parent strand to the daughter strand, so that now this pattern of methylation is present in both copies of the genome, so that when the cell divides, the two daughter cells both get identical copies of the genome and the epigenome, because that methylation pattern was copied. There are a different set of enzymes that are functional during development that can establish new patterns of methylation. So at very specific times in development in mammals, the methylation pattern is wiped clean, and the genome is clean of these methylation marks, and now these enzymes can come in and lay down new patterns. And so different patterns can be inherited from each of the parents. This is called imprinting. So some gene clusters become methylated during sperm production, others during egg production. So since the sperm and the egg have different patterns of methylation, that means that you've essentially marked which genome came from mom and which half of the genome came from dad, right? So now in the new offspring, both the mother's and the father's genomes are marked in different ways. This can lead to differential expression of maternal and paternal alleles in the offspring, right? If methylation is shutting down a region of DNA, and that region is only methylated on the paternal allele and not the maternal allele, that means that the paternal allele is going to be shut off and the maternal allele is going to be expressed, and that's going to be true for every offspring that's produced. When this happens, when one allele is shut down such that the other one is the only one that's expressed, this is called monoallelic expression of a gene, right? Monoallelic meaning just one of the alleles is being expressed, the other one is shut off. This is in contrast to what we typically think of as biallelic expression, right? Typically we think of when a gene is expressed in a diploid organism, that both alleles are going to be expressed in the same way, right? And so being homozygous or heterozygous matters. That would be biallelic expression. But when DNA is methylated in imprinting situations in which the maternal or the paternal cluster is methylated, we can have monoallelic expression of a gene cluster. So I want to use a specific example, and this diagram comes from a nice review by Delaval and Phil from 2004. And so the picture is a bit more complicated now, and in fact that's what we're going to be exploring in class. But this provides the general scheme of how the Igf2r locus works. You'll notice that on the top here in pink is the maternal part of the genome, and in blue on the bottom we have the P, or the paternal set of genes. Each of these boxes represents a different gene that you can see is labeled there in italics, as is our common nomenclature. So the large box here is the Igf2r gene. This is for insulin-like growth factor receptor, or insulin-like growth factor 2 receptor. And then we have SLC22A1, A2, and A3. So these are the genes. You'll notice that the genes are present on both alleles, and the arrows are indicating expression of those genes. So you'll notice that the Igf2r gene is only being expressed on the maternal allele, but we don't see that arrow on the paternal allele. The maternal allele is also expressed for each of the three genes in the SLC22A cluster. But the story is quite different on the paternal allele, where we see that Igf2r is not expressed. We don't see that little arrow there, but instead we're getting expression of this RNA called AIR. It's also called AIRN in our paper. We see expression of SLC22A1, but not A2 or A3. You'll notice those arrows are missing. So why do we get different expression of these four genes depending on the maternal or the paternal allele? It has to do with the methylation pattern. you'll notice that here it's the maternal allele that is methylated, and that is right over the ICR, which is a control region, imprinting control region. And so this imprinting control region is methylated on the maternal allele such that the heir RNA is not produced. Okay, so that heir RNA is not produced on the maternal allele, and so we get expression of all 4 genes. The father's allele is not methylated, and because it's not methylated, it's not shut down, and that means that we get expression of this heir RNA. Now, how does that cause the shutdown of Igf2r, SLC22A2, and SLC22A3? Well, that's a story that we're going to explore later on. It turns out that there's a different mechanism for shutting down these 3 genes. SLC22A2 and A3 are shut down in one way, and then what we're going to explore in this week's paper is how the Igf2r gene is shut down on the paternal allele. So hopefully that gives you some useful background information for this week's paper, sort of sets the stage, and I look forward to talking about the mechanism of this imprinted locus in class. I'll see you then.